Here's everything you need to know for Biology Paper 1. And the first thing I want you to remember is that eukaryotic cells are things like plant and animal cells. They have a nucleus, they have membrane-bound organelles. Whereas prokaryotic cells are things like bacterial cells. They do not have a nucleus, they have plasmids, they have chromosomal DNA, and they are much, much smaller than eukaryotic cells. And just remember that whilst they both can have cell walls, for example, plant cells have cell walls, animal cells don't. In prokaryotic cells, they do have cell walls, but bacterial cells, for example, have cell walls that are made up of peptidoglycan, which is something that antibiotics attack, which help to destroy the bacteria. Another thing I want you to remember is that when you're converting between millimeters and micrometers, you are multiplying by 1,000 and you're dividing by 1,000 if you're going from micrometers to millimeters. Make sure if you're doing any calculations tomorrow, you're doing all the calculations in the same units. Don't have one thing in millimeters, another thing in micrometers, because then you may get confused. Now, a lot of you guys will already know about the specialized cells, but I'm going to go through the ones which I think most people mess up on. Nerve cells or neurons are really long cells, and they have a really long axon insulated by myelin sheath, which is a fatty insulation layer, and they're really long so that they can reach the whole body. So they can send signals from your CNS, from your brain, to everywhere else around your body. The myelin sheath, which makes the axon kind of look like hot dogs, is actually an insulative layer which just allows for impulses to travel faster along those neurons. I also want you to remember that with sperm cells, they have something in the tip of the head, which is called the acrosome. The acrosome contains digestive enzymes to help break through the egg cell walls so that fertilization can occur. In the middle of a sperm cell, you have the midpiece, and that contains a whole load of mitochondria so that it can respire, so that it can power the flagellum, the tail. And of course, remember that the nucleus is found in the head of the sperm cell, and the nucleus contains half the chromosomes required, so 23 chromosomes, to create a zygote. I also have a video on phloem and xylem cells, so make sure you watch that if you're unsure about those. For microscopy, I want you to remember the triangle IM, where we've got image size, actual size, and magnification. And this is where what I said earlier about the units is really important, because if you use an image size in millimeters and you're calculating the actual size, that will also be in millimeters. So if the question asks you to give the actual size in micrometers, you've got to make sure you convert between millimeters to micrometers by timesing by 1,000. When we're doing microscopy, there's a couple of things I want you to remember. Firstly, we always start on the lowest objective lens, the lowest power, and then we get more zoomed in as we go on. When you're focusing on the subject, the thing in your slide, you want to make sure you use the coarse adjustment knob first, and then when you've got it and the image looks almost clear, then you use the fine adjustment knob and you just make it perfectly clear. When you want to view the image at a higher magnification, you've got to redo those steps again. So you've got to use the coarse adjustment knob again, the fine adjustment knob again, and get it to focus in on the image. Something that hasn't come up in a while is scientific drawings. Remember, when you're doing scientific drawings, there are certain rules you have to keep in mind. We learned that we only draw what we can see. So whatever you see through the microscope, that's what you can draw. If you can't see the mitochondria, don't draw them. We only use straight lines. We don't do any shading or coloring in. We label any features that we want to label with straight uncrossed lines, so don't start having lines which cross each other, making it all confusing. And as a rule of thumb, we always try to make sure that our drawings take up at least half the page or half the space that we're allowed to draw the drawing in. I've just recently done a video on aseptic techniques as well, so make sure you watch that if you want to learn about culturing microorganisms using aseptic techniques. And that's it for part 1. I'm going to release a part 2, part 3, and part 4. Make sure you guys like, send this to a friend, and see you guys on the next one.